Hello and welcome to Protein 101. Now in this PowerPoint we're going to look at the basic structure of proteins and basic biochemistry behind them. So let's get started straight away. First of all, about 50% of the organic matter of the cell is made up of proteins, and there are four main elements in the protein itself. That's carbon, hydrogen, oxygen and nitrogen, and we'll see also there's a very limited amount of sulfur. Functions of proteins, well, they can be structural, that is building the actual shape of the cell. They can be membrane carriers, that's for getting other chemicals across the membrane into the cell. They can be used as enzymes to speed up reactions, hormones or antibodies. They are involved in growth, that's in muscle growth and repair of tissue, and also they have metabolic functions. And there are approximately 10,000 different proteins in organisms. Now in terms of the building blocks, what are they made up of? Well, let's start at the very beginning. A monomer is a single unit, as we'll look at in a moment. Now the single unit, if you like, the basic building brick of a protein is an amino acid. If you get two amino acids joined together, they form a dimer, or what's called a dipeptide. When you get many amino acids joined together in a chain, you get what's called a polymer, or polypeptide. But let's look first of all at the monomer unit. Now this is the basic monomer of an amino acid. So amino acids join together to make proteins. So this is the basic building block. Now in this amino acid you've got the acid group here. The COOH is the acid group. You've got here an amino group. NH2 is an amino group, and it's this part here that's a variable group. These bits all stay the same in every protein. It's only this bit here that changes. Let's have a look now at the most simple of all the amino acids. This is glycine. Now glycine, remember the R group? Well, the R group here is simply H. That's all there is to it. Okay, so glycine is the simplest of all the amino acids. Now all amino acids, as I said, have the same basic structure. That's the amino group, the carboxylic acid group, but it's the R group that varies. Now the R group gives different properties to each individual amino acid. It can change the charge. It can make the amino acid hydrophobic. Hydrophobic means basically water hating, or it can be hydrophilic, which means water loving. So these are all properties that are given by whatever that R group happens to be. Now we as humans need 20 different types of amino acids. Now of these, 8 cannot be made in animals. They can't make them. These are known as essential amino acids, and we need them in our diet. Another thing is the body cannot store amino acids because the amino group is toxic if it accumulates. Amino, NH2, easily converts to ammonia, NH3, which is toxic, so they are broken down in the liver into urea, which you excrete, and this is called deamination. So deamination is a breakdown of these amino acids. Now here's the 20 amino acids that we need. The 8 in red are the essentials that we can't manufacture. Okay, so alanine all the way down to valine. Basic look at the structure. We're not going to get too much into it at the moment, but you can see here non-polar, non-ionizable at the top, polar, basic and acidic there. Now how do they join together? Well, they join together by what are called condensation reactions. So these amino acids join by condensation. If you know what condensation is involved with water, so it's about condensation on windows, for example. So a condensation reaction involves removing water to form a covalent peptide bond. This bond can be broken by the reverse process, by what's called hydrolysis reaction. And in hydrolysis reactions, what happens is that that water is added to it, it gains water, the bond is broken, and this is controlled by enzymes called proteases. We look at it in a diagram here. Here we've got two simple amino acids. Here we've got OH and H, and basic chemistry tells you those two make H2O, water. So by eliminating these two here to form water, you end up with what's called a dipeptide molecule down here. So here's a dipeptide molecule down here, and here is the peptide bond. This is a peptide bond here. So it can be reversed by adding water. It can be formed by taking away water. So you've got this cycle here of making it. Now if you look, we can do this at either end. So we can do it here, we can do it here. So basically you can end up with very, very long chains of amino acids. And here we've got a long chain. A polypeptide is basically a short protein. It's a short protein. You can see here we've got the peptide bonds being formed. Okay. Now there are 4 levels of complexity of proteins in terms of their structure. Where are they made? Well, proteins are made inside the cell. Inside every cell, proteins are made, and inside the cell you've got structures called ribosomes. Now ribosomes are found in the jelly part, the cytoplasm, and they are manufactured using something called mRNA. Now you may have come across DNA, which is deoxyribonucleic acid. Well, RNA is ribonucleic acid, and M stands for messenger ribonucleic acid. Now we're not going into any detail at the moment, but this is the essential bit of kit here that codes for what order the amino acids will go in. And what this does, it creates a chain of amino acids. As I say, this chain, fairly short, is called a polypeptide. Longer polypeptides are called proteins. And this is known as the primary structure. So the primary structure is a sequence of amino acids. Now different amino acids have different properties, so the sequence they're placed in will affect the actual properties of the polypeptide. And here we've got a polypeptide chain. These are all the abbreviations for the amino acids: glycine, valine, cysteine, glutamine, alanine, serine. They're all joined together here, okay, to form this chain. This chain is in fact a polypeptide. Here we've got a bond here between 2 sulfurs. Cysteine contains sulfur. Remember I said early on that sulfur is found in very, very few amino acids. Well, there's the sulfur forming a bond there, okay, and at one end you've got the amino part, the other end you've got the acidic part there. So this is a polypeptide chain. So when we form a polypeptide chain, all we're doing is sticking together amino acids, and these could be any of the 20. So this could be valine or whatever, the valine part. There are 20 different types of amino acid, so each position in the chain, any one of the 20 amino acids could be present. So this could be valine, cysteine, glutamine, alanine, and so on. Now even a chain as small as 4 amino acids, there are huge numbers of possible combinations. In fact, 160,000 different sequences if you had just 20 amino acids in all possible 4 amino acid combinations. Now most proteins have chains of more than 100 amino acids. They're pretty big molecules. Now the primary structure is simply how they're put together, that is valine, cysteine, glutamine, and so on. The secondary structure describes something about their shape. Now there's 2 basic shapes: there's coiling or folding. So they can be coiled here like this one here to form what's called an alpha helix, or they can be pleated like this one here. It's a difference in shape. That's called a beta sheet. They're held together by what are called hydrogen bonds. Here we've got it again, the secondary structure. This is the alpha structure here. Okay, you can see there the rotation of the amino acids. There's 10 turns of coil. Hydrogen bonds form between one amino acid and 4 places along the chain. This is the beta pleated sheet over here, very distinct difference in shape, but these are secondary structures. So remember, in proteins there's 4 structures. The primary is the basic sequence of amino acids. The secondary is whether alpha or helix. Now the tertiary structure, we start now getting more complex shape. It describes the 3D structure of the protein. Okay, we can have coils and pleats. The amino acids chains fold on themselves and they're held in place by different types of bonds. Okay, and notice there we've got 2 different chains there. Okay, 2 different chains. We've got a beta chain and alpha chain. These can be very, very long chains depending on how many amino acids they've got. You can picture this, you know those telephone wires that coil in on themselves and coil again and get rather irritating? Well, this is like the same sort of structure. It's a super coiling of these things here. Here again is the tertiary structure. Key features: they're held by hydrogen bonds, also they've got ionic bonds, disulfide bonds, and there are, remember I talked about this earlier, hydrophobic and hydrophilic interactions. Very, very important there in membrane structures. The top level is quaternary structure. So primary was the basic sequence amino acids, secondary alpha and beta structures, tertiary is coiling, and in quaternary structures more than one polypeptide joined together, often joined to an inorganic molecule such as iron, and this is called a prosthetic group. And examples of molecules that are quaternary structures are hemoglobin in your blood and also insulin. Hemoglobin is a globular protein and consists of 4 polypeptide or long protein chains. It's got 2 alpha chains and 2 beta chains held together with bonds, and each, as you probably know about hemoglobin, contains iron, and these iron molecules are called heme groups. This is a rather beautiful picture here, a model of a quaternary structure, and the last one we've got is the hemoglobin structure. So here we've got the 4 chains, 2 alphas, 2 betas, and we've got heme groups here, the iron groups of 4 of these heme groups to every molecule of hemoglobin, and those are the ones that pick up the oxygen. Okay, well that's a really rushed summary of protein structure, but I hope you now feel a bit more knowledgeable about proteins. Okay, so thanks for watching this video.